Today we're going to be talking about antibiotic-resistant bacteria. My name is Lena Ravetti, and I'm here with Long Trin and Ignacio Bermudez. And together we will talk about how bacteria can fight off antibiotics, ways in which they replicate, and the environmental consequences of this phenomenon. First and foremost, antibiotic resistance occurs when microbes evolve mechanisms that protect them from the effects of an antibiotic. Antibiotic resistance begins with a bacteria that acquires a mutation in their DNA sequence that codes for a protein which gives them a tool to fight the antibiotic. There are several mechanisms by which this tool can work. One way is by prevention. It can prevent the antibiotic from entering the bacterial cell to begin with. For example, gram-negative bacteria have an outer membrane that protects them from their environment, and these bacteria can use this membrane to selectively keep antibiotic drugs from entering. Another way is by pumping the antibiotic out through its cell wall. Some Pseudomonas bacteria can produce pumps to get rid of several different important antibiotic drugs. Next is by inactivation of the drug. The mutation can code for a protein that will change or destroy the antibiotic through an enzymatic reaction. Additionally, there's modification of the target. It can modify or destroy the antibiotic's target so that it may no longer bind to the antibiotic. For example, E. coli bacteria with the MCR1 gene can add a compound to the outside of the cell wall so that the drug colistin cannot attach to it. Lastly is by avoiding the target. It could give the bacterial cell a way to bypass the antibiotic's target completely, rendering the antibiotic useless. For example, some Staphylococcus aureus bacteria can bypass the drug effects of trimethoprim.
Bacteria can gain antibiotic resistance through spontaneous mutation and inheritance. However, it's most common that bacteria gain it through horizontal gene transfer, which could be through conjugation, the process in which genes are transferred from the donor to the recipient bacteria. Transformation, another mechanism where dead or decaying bacteria that possess the antibiotic resistant gene gets transferred over to another bacteria. This is the second mechanism. Lastly, transduction, the mech where a bacteriophage infects a bacteria and duplicates identical copies of itself. Some copies might possess the antibiotic resistant gene where it can affect another bacteria and give it resistance.
Antibiotic-resistant bacteria have had numerous impacts on human life. However, it wasn't extensively studied or defined until the late 19th to early 20th century. Various instances throughout human history have resulted in advancements in understanding and possible treatment to antibiotic-resistant bacteria in order to improve human life. In 1983, there was an outbreak that claimed the life of a 72-year-old woman in Arizona after ingesting raw milk. The infection was discovered to be caused by a multi-drug resistant bacteria called Salmonella typhimurium, which still remains a threat to human health if ingesting unpasteurized milk. Another instance in which antibiotic resistant bacteria have menaced human society was in the 1950s when a seemingly harmless innovation of enhancing feed for livestock with antimicrobial resistant growth promotants, or AGPs, was shown to improve feed to weight ratio. Although the idea proved lucrative, an unforeseen outcome was discovered by microbiologists and infectious disease experts that farmers using AGPs with their livestock feed had more antibacterial resistant bacteria in their intestinal biomes than farmers not using AGPs. Up to its discovery in 1928 by Alexander Fleming, penicillin was a revolutionary treatment to infections in hospitals that greatly reduced mortality as opposed to the current time period's treatments, but was short-lived when in the early 1940s, penicillin-resistant Staphylococcus aureus emerged in London hospitals. As mortality rates began to rise attributed to the newly resistant microbe, methicillin was discovered to be the new treatment, which gave way for a more deadly and contagious hospital infection, which is methicillin-resistant Staphylococcus aureus, or MRSA.
To leave it off in a more positive note, future research needs to be conducted in order to fight off antibiotic resistant bacteria. These bacteria, like the ones mentioned before, are evolving every day, and with each generation that passes, they become stronger and more of a threat. It is our jobs as future scientists in the making to be part of these future researches so we can make a better Earth to live in.